Hi everyone. So let's begin our new session of lipid metabolism. Hope you have watched our previous video. And today we are going to discuss about certain disorders, that is atherosclerosis, fatty liver, obesity. Also I have included part B of our chapter, that is amino acid metabolism. In that we are going to dealing with general reactions of amino acid metabolism, then transamination, deamination, decarboxylation. So let's begin. Atherosclerosis. So atherosclerosis means it's a disease condition that inside our arteries the plaque gets built up. That the disease condition is called atherosclerosis. Plaque means it's a fat or cholesterol or other substance that is found in our blood. So as a time going, the plaques will harden and they will narrow in our arteries. So this results in oxygen rich blood flow will get stopped to our organs or other parts of our body. So this may even result in death, heart attack, stroke, etc. So these plaques of atherosclerosis can cause three main kinds of cardiovascular disease, that is coronary artery disease, cerebrovascular disease, peripheral artery disease. So first one, that is coronary artery disease. That means the stable plaques in the arteries cause angina. Angina means chest pain. Exit. Then this plaque will get rupture and clotting occurs. So it results in death, heart attack, etc. Next is cerebrovascular disease. Cerebrovascular disease means ruptured plaques in the arteries causes strokes. These ruptured arteries in coronary artery means the stable plaques has been formed, after that only it get ruptured, but here there are already the arteries get ruptured and this results in stroke and finally it results in brain damage. Then third one is peripheral artery diseases. Peripheral artery means arteries of legs will get narrow, that is in results with plaque. So it results because of poor circulation. So while we are walking we feel pain on our legs and it will take more time for wound healing, etc. So causes, how this disease get caused, let's see. First one is smoking. If you smoke more it is dangerous, and due to high cholesterol, high blood pressure and diabetes, obesity means overweight, and stress. Then not we are eating not fruits and vegetables, then excess alcohol intake, and we are not taking proper exercise each day. Next is treatment means lifestyle changes means we have to follow a healthy diet, exercise and smoke less. Then so this results in good life. So there is no chance of these diseases. Then medication means taking drugs for cholesterol, blood pressure. Bypass surgery means the surgeon will take an healthy blood vessel from our leg. Then they use that vessel to bypass segment blocked by this atherosclerosis. So that is about atherosclerosis. Next is fatty liver. Fatty liver. So we all know that the normal concentration of lipids in liver is only 5%, and it is a storage organ for fat, that is adipose tissue. Then lipids means especially triacylglycerols. They will accumulate more in our liver and they resulting in fatty liver. And in normal liver, that is Kupffer cells, they contain lipids, that is in the form of droplets. So these droplets will found in cytoplasm of hepatic cells and they results in impairment in metabolic functions of the liver. And this fatty liver is associated with fibrotic changes and cirrhosis. So it is mainly due to two conditions, that is increased synthesis of triacylglycerols. Next one is impairment in lipoprotein synthesis. First one, increased triacylglycerol synthesis. So here the fatty acids, that is from adipose tissue, will mobilize and they will influx into liver more than their utilization. They directly move to the liver. So this leads to the more production of triacylglycerols and this is accumulated by our liver. That some disease conditions like diabetes mellitus, starvation, alcoholism, high fat diet, these are all related to this. And alcohol, they will inhibit fatty acid oxidation and they will promote the fat synthesis and their deposition. Next one is impaired synthesis of lipoproteins. That means the synthesis of very low density lipoprotein takes places in liver. So this VLDL, that is very low density lipoprotein, it will require phospholipids and apoprotein B. So this fatty liver may be caused by impaired lipoproteins. So that is mainly due to defect in phospholipid synthesis. Also there's a defect in apoprotein formation. And finally the secretion or the formation of lipoprotein. And among these three causes, the fatty liver is mainly due to impairment in phospholipid synthesis. So actually it is associated with dietary deficiency of lipotropic factors. Lipotropic factors means example choline, betaine, inositol, etc. Then deficiency of essential fatty acids also results in decreased formation of phospholipids. And also the excessive consumption of alcohol competes with these essential fatty acid and they will impairs the phospholipid synthesis. For example, certain chemicals like carbon tetrachloride, chloroform, phosphorus, etc., they will inhibit protein synthesis and causes fatty liver. It is mainly due to the synthesis of apoprotein B that is required for the VLDL, that is very low density lipoprotein. And this lipoprotein synthesis, therefore, and their secretion need ATP. Sometimes there will be less availability in ATP. So at that time it will impair the lipoprotein formation. Okay. Next is another reason, that is endocrine factors. endocrine factors means, for example, certain hormones like insulin, thyroid hormones, adrenocorticoids, they will promote the deposition of fatty liver, so they results in fatty liver. Next is lipotropic factors. Lipotropic factors means, we have already told that examples choline, betaine, methionine, these are the lipotropic factors. So what they are doing, they also causes fat deposition, and it is accumulated in our liver and results in fatty liver. And how they are acting, let's see. That is choline or inositol install. They are the components of phospholipids, and they are very important for the synthesis also. And other lipotropic factors, they may be directly or indirectly related to the transmethylation reactions. So then the synthesis of choline, they results in fatty liver. This is mainly due to the deficiency of choline. So as a result, insufficient amino acid will be supplied. In other words, we can say the non-availability of methyl groups will lead to fatty liver. Next is choline deficiency and fatty liver. So it is mainly due to certain reasons, same reasons only, that is phospholipid synthesis and the formation of lipoprotein membrane, impaired formation of lipoprotein membrane, and reduced synthesis of keratin, that is due to the insufficient supply of methyl groups, and also impairment in fatty acid oxidation. And that's about fatty liver. Next is obesity. Obesity means it is a medical condition, that is excess fat has been accumulated, that is called obesity. So obesity is generally considered, that is considered by their body mass index, that is BMI, that is measurement obtained by, we are dividing person's weight by the square of person's height. So thus we will get the BMI. Then obesity increases various disease and other conditions like cardiovascular diseases, type 2 diabetes, sleep apnea, certain types of cancer, osteoarthritis, and depression, etc. Then what are the causes? Causes means excessive food intake results in overweight and lack of exercise, and some cases, very rare cases, it comes from genetics. Also any other medical reasons or any psychiatric illness. It is all rare cases, and most familiar cases is excessive food and lack of exercise only. Then management means we have to maintain proper diet, also we have to do our exercise regularly. This is only way to reduce our weight. Then most effective treatment that for obesity is bariatric surgery. So it is these types of procedures include laparoscopic, adjustable gastric banding, etc. And the surgery is for severe obesity, and that's for obesity. Next is amino acid metabolism. That's our second part of our chapter. Amino acids. We already discussed what is amino acids and who produces amino acids, etc. In our first chapter, we have discussed. So in that first topic is general reactions of amino acid metabolism. That is, there are many reactions. There is transamination, deamination, decarboxylation, etc. So amino acids, they'll undergo what common reactions like transamination, that is followed by deamination, and the results in the liberation of ammonia. So this amino group of amino acids is mainly utilized for the formation of urea, which is excretory and the product of protein metabolism. Then the amino acids carbon skeleton, that is first converted into keto acids, that is by transamination. So they having certain factors. They are mainly used to generate energy. Also they used in the synthesis of glucose and formation of ketone bodies, the production of non-essential amino acids. So transamination. Transamination means the amino group, that is the amino acid will transfer amino group to keto acid. That is known as transamination. See, you can see this picture representation. From this only you will get understand what is transamination. Nothing but an amino acid that is transferring amino group to keto acid. That is called a transamination. So you can see that this is amino acid and they getting converted into keto acid. So here second amino acid they are getting converted into keto acid using this one amino transferase, and here it is PLP, that is pyridoxine phosphate. Next is silent features of transamination. See, for all transaminases, it requires PLP, that is pyridoxal phosphate, and a coenzyme. That coenzyme is derived from vitamin B6. And certain special transaminase exist for each part of each pair of amino or keto acids. For example, that is aspartate transaminase and alanine transaminase. So they are the significant contribution for the transamination. It's the best example. Then there will be no free ammonia liberated. Only during transfer only the ammonia is liberated. Next, it is reversible reaction. And the transamination is very important in redistribution of amino groups. Also non-essential amino groups for the requirement of the cell. And they involves both catabolism and anabolism. Catabolism means degradation, anabolism means synthesis. Then the transamination diverts the excess amino acid towards the energy generation. For energy production, they diverts the more amino acids. Then amino acids will undergo transamination to concentrate nitrogen in glutamate. Do you know what is glutamate? Glutamate means it is an amino acid. When they undergo oxidative deamination, ammonia is liberated for urea synthesis. And all amino acid except lysine, threonine, proline and hydroxyproline participate in transamination. And transamination is not restricted towards alpha amino acids. For example, 8 amino group of orthonine is transaminated. Next is serum transaminases. These are very important for the diagnosis and prognostic purposes. Next is mechanism. This is a mechanism of transamination and it occurs in two stages. First one is we are transferring amino group to coenzyme, that is pyridoxal phosphate, to form pyridoxamine phosphate. Next is this amino group of pyridoxamine phosphate is transferred to keto group to produce a new amino acid, and an enzyme, that is with PLP it is generated. So that is, while seeing this picture representation you will get a clear cut idea. Next is deamination. Deamination means the removal of amino group from amino acid. That is called a deamination. There are two type of deamination, that is oxidative deamination and non-oxidative deamination. So let's discuss oxidative deamination. Oxidative deamination is nothing, it is a liberation of free ammonia from amino group of amino acids, and it is coupled with oxidation. That's why we called it as oxidative deamination. This will takes place mostly in liver and kidney. And what is the purpose of this deamination is we are providing ammonia for urea synthesis and also keto acids for a variety of reactions, and they also include energy generation. For example, the role of glutamate dehydrogenase. Next is non-oxidative deamination. Non-oxidative deamination means there are many categories, that is one is amino acid dehydratase. Here serine, threonine, homoserine, these are hydroxy amino acid and they are undergoing non-oxidative deamination, and this reaction is catalyzed by PLP. PLP, and it results in keto acids. Next is amino acid desulfurhydrase. Here sulfur amino acids are used. Example cysteine, homocysteine, and they will undergo deamination and results in keto acids. Next is deamination of histidine. Here histidase enzyme will act on histidine and liberate ammonia, and they will liberate ammonia, and this is deamination. Next is decarboxylation. Decarboxylation, from the name itself we'll get it is a removal of carboxyl group, and they releases carbon dioxide. There is removal of carbon dioxide from amino acid by the formation of amines. This is the example. So this is the amines, and the removal of carbon dioxide we'll get amine. Usually the amines will have high physiological activity. So these are the examples of decarboxylation. That is triosine to trimine, histidine to histamine, serine to ethylamine, glutamic acid to GABA, lysine to cadaverine, arginine to augmentin, etc. These are all examples for the decarboxylation. And you don't want to write the reactions, you can study these reactions only. Wait, these are the examples. Triosine is converted into triamine. Histidine is converted into histamine in the presence of histidine decarboxylase. Next is our final topic, that is metabolism of ammonia. So ammonia is it is liberated from the metabolism of amino acid or other nitrogenous compounds. So during particular pH, ammonia will exist as ammonium ion. So first one is formation of ammonia. That is for the formation of ammonia we require amino acids. That is by the reaction transamination and deamination. That is amino acid of purines or pyrimidines, or by the action of intestinal bacteria on urea. This is how the ammonia is formed. Next is transport and storage of ammonia. Transport and storage means the regular or a constant production of ammonia from various tissues Their concentration in circulation is surprisingly low, that is 10 to 20 mg per dl. So this is because the body has sufficient mechanism on ammonia transport and its utilization. So this transport of ammonia between various tissues and liver occurs in the form of glutamine or alanine, not as a free ammonia. And alanine is very important for ammonia transport, that is takes place in muscle to liver, that is by glucose alanine cycle. It is clearly written in your textbook. Next is disposal of ammonia. So we have already produced ammonia, that is then it is transported, and it is storage also we have discussed. Then is we have to disposal ammonia, that is organisms during their evaluation had developed many other mechanisms for the disposal of ammonia in our body. So that is first one is aminotelic. Aminotelic means it is aquatic animals. They will dispose ammonia into water. Then is uricotelic. Uricotelic means ammonia is disposable in the form of uric acid, birds and reptiles, etc. Then ureotelic. Ureotelic means mammals will convert ammonia to urea. Urea is non-toxic and they are soluble also, hence we can easily excrete. Finally, that is toxicity of ammonia. Toxicity means blood ammonia, that is ammonia will elevate it. Elevated means increases. So this concentration is harmful to our brain. So this will accumulates our body, and they results in slurring of speech and blurring of vision. Blurring means we cannot see properly and we cannot speak properly, and this results in streamers, and they even leads to coma and finally to death. So this reaction is catalyzed by glutamate dehydrogenase. So it will clearly explains the toxic effects of ammonia in brain. So that is how it is happening. Ammonia is accumulated and it is shift to the right direction with more glutamate formation. So thus the more glutamate is formed, soonly the utilization of alpha ketoglutarate takes places. Alpha ketoglutarate means it is a very important enzyme in citric acid cycle. So their level will, when it increases, it will impairs the citric acid cycle, and net result is production of ATP will be reduced. So the toxic effect of ammonia on brain will get more. So this results in impairment in ATP formation. So that is very dangerous situation. So citric acid cycle we didn't discussed. Later classes we will come across that cycles. So that's for today. Thank you.